So welcome guys to photosynthesis. So a lot of people have a lot of photosynthesis, but I think that you'll see that photosynthesis is actually biochemically just really cool. It will also kind of give us that balance between the cellular respiration that we looked at in Chapter 9. Chapter 9 was all about taking electrons from carbon. In photosynthesis, we'll be able to get those electrons back. So let's take a look at the sort of relationship between mitochondria and chloroplasts. So a common misconception is that plants only contain chloroplasts and don't have mitochondria, whereas things like animal cells have only mitochondria. While it's true that animal cells only have mitochondria, plant cells require both chloroplasts and mitochondria. They're able to put electrons onto carbon through photosynthesis, but in order to get the ATP they need to do cellular work, they also need cellular respiration. So we do need kind of a balance. In cellular respiration, which again was in our mitochondria, we were taking electrons from carbon, putting them through the electron transport chain, and getting ATP. Along the way, that carbon that we oxidized was released as CO2. You can think of CO2 as kind of like a drained battery. We can't get any more potential energy out of this to power our daily living. So this is kind of like a drained battery. So you can imagine that over time, we would just be, eventually the world may be completely out of batteries if we have no way to recharge them. So photosynthesis is our answer to that. So we want to take those electrons back and put them back onto carbon. So we're going to use the energy from sunlight because we saw that it's not super easy to pull electrons away from oxygen. We're going to use the light energy to put those electrons from oxygen back onto organic molecules, aka sugars is what we're thinking about. So at that point, we could put these back into cellular respiration, but those batteries are recharged. So photosynthesis will allow us to reduce carbon. Cellular respiration was oxidizing carbon. So let's compare these net reactions. Well, so with cellular respiration, we know, again, that we take sugar, oxygen, it's going to be our electron acceptor. We get water, carbon dioxide, and energy, in principle, in the form of ATP. In photosynthesis, we require energy. So from the start, this can be energy from the sun. So we're going to be converting that to potential energy to power part of the photosynthetic reactions, but our initial energy that we're getting, this is energy from the sun. We'll require some water. We'll see where that happens. And carbon dioxide. This is that part where plants take up carbon dioxide and give us oxygen, and then we also get sugar. So that's all good. We're going to see how this occurs in our next couple of slides. So here we're looking at the structure of a chloroplast. So we know that photosynthesis happens in chloroplasts. This is the outer membrane. This is the inner membrane. We're not going to be hanging out at that inner membrane today. So instead, chloroplasts use thylakoids to get their surface area. So these are just sort of like membrane-bound discs. And so these thylakoid membranes are going to serve a similar analogous purpose to the inner membrane of mitochondria. This is where we're going to see our photosynthetic machinery living. So our light reactions are going to take place at this thylakoid membrane. We're going to see some stuff going on inside the thylakoid. This is called the thylakoid space. And then we'll see some things happening out here, outside the thylakoid. This is called the stroma. So we'll see a couple of different things going on. So our light reactions, we're really going to see a lot of stuff taking place at the thylakoid for this part of it. The light reactions, we're going to need energy, again, from the sun and water, sunlight and water. This will get us part of what we need to do the second half of photosynthesis. So this converts energy from the sun into our potential energy, chemical energy, and redox potential. The NADPH is similar to NADH that we saw in cellular respiration. So it is an electron carrier. This is a prize right here because this holds the electrons that we're going to ultimately put back onto carbon. So the second part is we're going to need some carbon, and this is going to take place through the Calvin cycle. So ATP plus NADPH that we got converted from light energy, add this to carbon. This is going to allow us to make sugar. So when we balance that reaction, we get the net photosynthetic reaction that we saw in the previous slide. So let's get started with our light reactions. So light reactions require light. It's actually true that the Calvin cycle requires light as well, just because it's dependent on the products from the light reactions. However, light reactions are very directly dependent on photons, so we'll see how that works. So what we're looking at here is going to be the membrane of a thylakoid. So outside is the stroma. This is just the inner space inside the chloroplast. Down here, this is the thylakoid space. So this is going to be the actual inside of that disc that we just looked at. So we can see some things going on here. This is our light reaction machinery. So the light reactions are going to be dependent on two complexes called photosystems. Photosystem 2 is actually going to be the first one we encounter. This is actually the second one discovered, so that's where it got its name. It's also called P680, which is based on the chlorophyll that we'll look at in a second. The second photosystem that we'll encounter, called Photosystem 1, or P700, will take place a little later in this mechanism. We're also going to have sort of an abbreviated electron transport chain. We've got some mobile carriers here, plastoquinone, plastocyanin, and viridoxine over here, mobile carriers, and the cytochrome complex. Hopefully we're comfortable with cytochromes at this point after our journey through the electron transport chain of mitochondria, and we know that cytochromes are going to be good places for electrons to travel down. So we've got an electron transport chain, and then finally ending up at an enzyme complex called NADP plus reductase. So I hope that you're getting kind of an image of what that complex might do, but this is going to allow us to actually get electrons, and this is going to be exactly where they go back onto carbon. So this is going to be where we're going to be taking electrons, they're going to go onto carbon and bringing them to the Calvin cycle. So let's take a look at photosystems. So these are worth investigating a little bit more closely. So what is a photosystem? So photosystem, all this purple part is going to be kind of like a protein structural complex, and embedded in these complexes are chlorophyll molecules. So we'll be able to look at the structure of chlorophyll on the next slide, but for now let's just enjoy the structure of the photosystem in general. So there's chlorophyll and other pigment molecules embedded all throughout these light harvesting complexes sort of surrounding the photosystems. Their job is 100% just to capture light. So we'll get back to them in a minute. Let's visit the center of this photosystem. So right here in the center, these are just chlorophyll molecules. Specifically chlorophyll A, there are a couple types of chlorophyll. That's not so important to what we're trying to get from today's video, but these chlorophyll molecules are called special chlorophyll molecules or special pair. So they're called the special pair not because the chlorophyll is inherently any different than some of the other chlorophyll out here, but because of its location in what's called the reaction center. So this is going to be where all of the action takes place. So we've got special chlorophyll pair, And up here, we're going to have what's called a primary electron acceptor. That's going to do exactly the job that you think it's going to do once we get electrons moving in the system. That's going to allow us to pull electrons and funnel them into the electron transport chain of the thylakoid. So we've got photons coming in. We're going to have special chlorophyll pair in the center, and then the primary electron acceptor. So let's take a closer look at chlorophyll, and then we'll see some of these photosystems in action. So just because I think it's useful to see what these molecules look like, chlorophyll A, here pictured, and there's chlorophyll B, and there are other pigments as well. They absorb red and blue light primarily. Each different type, has a preference for slightly different wavelength. And the thing about this is constructed of a porphyrin ring and a hydrocarbon tail. The whole job of this is to be able to absorb energy from the sun and have an electron that becomes excited once that occurs. So let's go back to our photosystem and see why that's going to be important. So when photons hit the chlorophyll in photosystem 2, so here comes the sun. Let's add some photons. Chlorophyll's electrons are excited in such a way that they don't move. They don't actually move from chlorophyll. Instead, they just raise an energy level, and this energy is transferred from chlorophyll molecule to chlorophyll molecule to chlorophyll molecule. So it's really important to kind of internalize that at this point, no electrons are moving. This is simply energy being transferred from one molecule to the next, kind of like if you're in a crowd doing the wave. So you're not really leaving, you're just raising up for a second temporarily and then passing that on to your neighbor. So all of that's fine until we reach this special pair. So now this energy reaches a special pair, and the electrons in the special pair, they raise up an energy level, but because of their proximity to the primary electron acceptor, this primary electron acceptor can actually grab these electrons. So it's kind of like, oh, I threw these up and now somebody caught them. So they don't return back down. The energy was transferred, and here electrons have physically moved. So it's kind of like an energy wave until it gets to the center where electrons actually move. Okay, so you can see that the electron transport chain's coming up. So hopefully you've got a good idea of what's coming next. Okay, so I've kind of colored this special pair black because now they've lost electrons. We'll get back to this in a second. For now, let's follow the path of these electrons. So they'll travel through the electron transport chain, plastoquinone, it's a mobile carrier, through the cytochrome complex, plastocyanin, which will carry the electrons to photosystem 1. So that's the immediate journey. We've got a little bit left after that, but we'll take a break right here. So here go the electrons through all of these complexes, taking a break on photosystem 1. When they get to photosystem 1, photons again cause these electrons, energy waves, to leave the special pair. They're grabbed by this primary electron acceptor, travel on this mobile carrier, ferroredoxin, and then ultimately to Nadp plus reductase. So I'm going to take a break right here. Okay. So at Nadp plus reductase, we're going to be doing exactly what it sounds like we would be doing, and we're going to be reducing Nadp plus. So here's a molecule of Nadp plus. This is going to be its oxidized form. It would like to be able to carry some electrons. Luckily, we had some right here on our reductase. So we're going to end up taking those electrons, putting them onto Nadp plus, to form NADPH. So this is going to be the carrier that takes these electrons to carbon. So we've pulled electrons for right now, kind of from this special pair, but we'll see what's going to happen here in a minute. These electrons are going to the Calvin cycle. That's one of the ingredients that we needed. So these electrons will be what we actually use to reduce the carbon. So we're ready to do that. Let's take a look now at this special pair that lost its electrons. So how do we get more electrons? It's not as if these have infinite electrons that they can continue to donate. Those were the only ones, the electrons that we previously saw, those were the only ones that they could potentially give. So where do we get more? Well, it turns out that as these electrons leave, they leave sort of an electron-shaped hole in the heart of these chlorophyll molecules, and this special pair that's now missing its electrons is more electronegative than even oxygen. This is one of the most electronegative things on the planet. This is like an even bigger bully than oxygen. So we're going to take those electrons back from oxygen. So if you remember from the last cellular respiration video, electron transport, we combined 2 protons plus 2 electrons and 1/2 an O2 to make water. We're essentially going to do the reverse here. So what we're going to see is that we're going to pull electrons from water. The electrons can be replaced onto the special pair. We end up with 2 protons and 1/2 an O2. This 1/2 an O2, this is a pretty fast process. It will immediately combine one oxygen with another to form molecular oxygen. So this is where the oxygen comes from when we talk about plants producing oxygen for our world. This is exactly where this happens. This is also called water splitting. So we split water, the electrons pulled from oxygen, and they're back on the special pair, but as soon as another photon hits, they're ready to go. So this is a pretty continuous process. So just to kind of recap and see the whole thing, just sort of the path. Again, Photosystem 2 has the capability to split water. So that's where we get our electrons. Energy from the sun, pop those electrons up an energy level where they are grabbed, sent through the electron transport chain, and at our Photosystem 1, energy level pop back up again from sunlight's energy onto ferredoxin, and then we're able to reduce NADP plus to NADPH, those 2 electrons, ready to go to the Calvin cycle to be put on carbon. So here, again, Photosystem 2 is the only photosystem that can actually split water. Photosystem 1 does not do this. So this is just kind of the recap. So you can see there's some ATP down here, and you may be thinking, man, we needed that for the Calvin cycle, but all we've made is this NADPH. So let's take a look at that. So you may have gotten an idea that the cytochrome complex is going to be involved here. That's the way that it worked in mitochondria. So we got our NADPH over here. It went to the Calvin cycle. The ATP is formed because this cytochrome complex pumps protons just like the ones in the mitochondria. It will pump them into the thylakoid space inside that membrane disc. So this is going to build up a really nice proton gradient, concentration gradient inside the thylakoid space so that we can do ATP synthesis. So somewhere else in the thylakoid membrane, so note that this is the stroma, the lighter color, the darker color is the thylakoid space. Let's kind of change our view for a second. Now we can say that we're somewhere else, maybe at the bottom of this thylakoid. The thylakoid space is at the top. We've been pumping protons in from above. And here's our ATP synthase. So chloroplasts have their own ATP synthase too, so they can do this work as well. We're going to see that protons travel around, just like they did in mitochondria. This is going to give us the energy to make ATP. So this is where we get our ATP from our light reactions. And if you think about the setup, this is pretty sweet. So not only are we getting the electrons we need to put back on carbon, but we're kind of using them in the process to get the ATP that we're going to need for this actual reduction process. So I think this is actually just a biochemical marvel. If you really think about it, it's got some of the coolest things going on. We had those special pair in photosystem 2, which is like the most electronegative thing that you're probably going to really deal with. And then we've got this sort of dual use, killing two birds with one stone kind of use for the electrons. We're going to put them back on carbon, and we're making some ATP. So we can see kind of the whole thing put together. Again, we're looking at the thylakoid, inside the thylakoid space. This is the path the electrons took. Okay, so we've talked about this. They're on NADPH now to the Calvin cycle. And we've pumped some protons in, used them for oxidative phosphorylation to make ATP, and that's headed to the Calvin cycle as well. So let's check out more about the Calvin cycle. So the Calvin cycle is going to be where we actually take our carbon, our CO2, remember this is that drained battery, and to charge it, we need to put electrons back on it. That's where this NADPH is going to come into play. Here, we're going to be using energy in the form of ATP. So we're able to convert energy from the sun into the chemical energy that we need to put carbon back the way that it was before we metabolized it. Okay, so let's take a look at the Calvin cycle. We're not going to spend as much time here as we did in cellular respiration looking at all of those reactions. It's really complex, but what will be important is just to keep track of the number of carbons that we're dealing with and which phase that we're in. So the Calvin cycle has 3 phases. The carbon fixation phase. This is phase 1, and this will be where we actually see carbon enter the cycle. Our second phase will be the reduction, or putting the electrons back onto carbon. And the third and final phase will be regenerating the acceptor for carbon dioxide. Okay, so that will be our 3 phases. Let's start with phase 1. So phase 1 is carbon fixation. This is where we take our carbon dioxide and stick it into the Calvin cycle. So this is not exactly like the Krebs cycle, but it's got some parallels. So this is going to be a cycle where we have an acceptor molecule coming around the back end, and we're going to be adding carbon in to create new molecules. So let's take a look at this back end molecule. So this is called Ribulose Bisphosphate, or more affectionately, RibP. So RibP is fine. This is an acceptable name. So we've got molecules of RibP, and each RibP molecule has 5 carbons. So 5 carbons, and we've got 3 of these molecules coming around the back. Okay, so this is going to be 15 carbons. So just kind of keep that in your mind. For the input, we've got carbon dioxide, which has 1 carbon apiece. We've got 3 of those. 3 carbons in. So that'll be the thing you need to tell yourself throughout this whole process, 3 carbons in. Okay, when we get to the output, we'll see that it lines up. So coming around the back, we've got 3 molecules of ribulose bisphosphate, each of which has 5 carbons. 3 molecules of CO2, each of which has 1 carbon. We're going to combine those together to ultimately get 6 molecules that are 3 carbons each. Okay, and that seems weird. We're going to look at the reaction on the next slide, but this is 18 carbons. 6 times 3 is 18. This is adding up. We had 15 from the RibP, 3 from the carbon dioxides. That was 18. So here all of our carbons are accounted for. They're just kind of in a weird form. So this reaction is catalyzed by an enzyme called Rubisco. So I won't have you learn most of the enzymes necessarily. in the Calvin cycle this time, but Rubisco is a really important one, and it's actually one of the most abundant proteins on the planet. So photosynthesis is just like, I mean, it's just really one of the coolest processes, okay, and it's got a lot of really important things going on. So let's take a closer look at this addition of carbon dioxide to a RuBP molecule to get this 3-phosphoglycerate. So here's our carbon dioxide, 1 carbon here. Here's our RuBP, 5 carbons here. We add it together to get an intermediate that has 6 carbons. Okay, so you don't have to like learn the structure, but just, you know, absorb. It's got 6 carbons. This is not a stable intermediate. This is really unstable, so it pretty much immediately busts in half. So you can kind of almost see where it happened. And we now have 2 3-carbon molecules. So that makes sense. 1 carbon dioxide, 1 RuBP. These are 6 carbons. We make the 6-carbon molecule, then bust it in half. 2 3-carbon molecules. Again, when we looked at phase 1 in the previous part, we're dealing with 3 molecules of each. So we ended up with 18 carbons here, or 6 molecules of 3-phosphoglycerate. Okay, so the second phase that we're entering is the reduction phase. This will be the phase where these electrons enter back in. You can see that that's the case because our NADPH is here, and we're going to oxidize it. We're going to take those electrons off of it, drop it back onto some of this carbon. Okay, so again, we're not looking at this in as much detail as we did cellular respiration, but you can see there's some stuff going on here anyway. So this is going to require energy. We have our 6 ATP. As we move through the cycle, we're here still 6 3-carbon molecules. This is 1,3-bisphosphoglycerate. And between 1,3-bisphosphoglycerate and glyceraldehyde 3-phosphate, we add these electrons back on so that we have a more reduced form of these sugars. Okay, so you don't, again, have to know exactly what happens here, but keep track of that the electrons are back on now, and notice how many carbons we have. We still have 6 3-carbon molecules. That's still 18 carbons. But check it out. We're about to drop one out of the cycle. This is 1 3-carbon molecule. Okay, so this is one of the G3Ps. We had 6 of them. We drop one out. We're going to see that we continue with 5. Okay, so we had on the back end 15 carbons plus 3. 18 carbons, 18 carbons, 18 carbons, drop 3 out, we'll be back to 15 for that last phase. So the actual direct product of the Calvin cycle is G3P. Now this can be used to build glucose, sucrose, other sugars. It can be used to make structural carbohydrates like cellulose, or it can be eventually metabolized for energy for the plant in the mitochondria, but this can be really used eventually for anything that you need it to be. So, but that'll be the direct form. 3 carbons in, 3 carbons out. So you needn't stress your brain about this or do any kind of crazy math. You put 3 in, you get 3 out. That's pretty much the deal. And since the 3-carbon molecule is the output, you have to put 3 in so that you can get 3 out. This is not full carbon alchemy. This is just 3 carbons in, 3 carbons out. Okay, so let's get that final phase. This is going to be our regeneration phase. So again, we dropped one of these G3Ps out. We continue with 5 more. We're just going to rearrange them. We're going to use some ATP. It's pretty clear we're probably going to drop one of these phosphates on the ends. It's fine. You don't really have to know, but we are going to use some ATP here. And we end up rearranging this so that instead of 5 3-carbon molecules, we end up with 3 5-carbon molecules of RuBP again. So this is just a continuous cycle. But really, just think about the phases: carbon fixation, reduction, regeneration of the acceptor, and keeping track of the carbons. 3 carbons in, 3 carbons out. So we've used ATP that we generated in the light reactions, and we've used NADPH to get those electrons to put back onto carbon. So the Calvin cycle, again, we fixed carbon into an existing sugar molecule, and we added the electrons back onto it, reducing it, leading to the formation of G3P, which is one of the things that was our outputs. Once we've done that, the starting sugar is regenerated, is rearranged so that we can get back to that Ruby P. So again, just to kind of recap the balance, our light reactions, we used photons to harvest electrons back off of oxygen. So from water, okay, where really, even though it's water, the oxygen was hogging the electrons. We took those back. Okay, the oxygen is now kind of on its own, okay, with left some protons floating around. This is the oxygen that plants give off. In those light reactions, we formed NADPH, and we used our oxidative phosphorylation to get ATP, fed these into the Calvin cycle, where we're able to fix carbon, grab it from the carbon dioxide form, put electrons back onto it, and then we're able to regenerate that. Okay, NADP plus that's used up can go back to the light reactions. Similarly, ADP can go be recharged in this reaction as well. So we've talked about the light reactions and what we can get from it. We've talked about the Calvin cycle and what we can get from that, how they work together, and we've talked about the balance between chloroplasts and mitochondria and cellular respiration and photosynthesis. So a lot of people like to focus only on cellular respiration, but we really need the other half, the other side of this coin, the photosynthesis, to keep that carbon kind of recharged, ready to go, ready to give us more energy.